Welcome back to Microbiology. In this video, we're beginning Chapter 10, the biochemistry of the genome. In this first video, Part 1, we'll focus on Section 10.1, how microbiology has been used to discover the secrets of life. And Section 10.1 traces key experiments that revealed DNA as the molecule of heredity and set the stage for modern genetics. Before the 1900s, many scientists believed in the blending theory of inheritance. So offspring were thought to be an average of their parents. This means that children kind of showed an intermediate physical appearance. Continuous variation in traits like height seemed to support this. So if a tall parent and a shorter parent mate and produce children, their children were often in between those heights, in between their parents' heights. But as we'll see, microbiology experiments overturned blending theory and established that traits are inherited as discrete units. These are what we now call or know as genes. In 1869, Friedrich Miescher, a Swiss physician and chemist, isolated a substance from white blood cell nuclei that he called nuclein or nuclein. Richard Altman later renamed it nucleic acid after finding that these were acidic molecules. And then Kossel later identified the bases A, G, C, T, and U, and we'll talk more about these later. These discoveries laid the foundation for molecular genetics. Gregor Mendel, an Austrian monk and botanist, worked with pea plants in the 1800s and was able to show that traits are passed intact through discrete units of inheritance. And he also demonstrated the existence of dominant versus recessive traits. His work, though, was ignored for decades and only rediscovered later on, becoming the basis of modern genetics. So this is a little bit beyond the scope of our microbiology course, but just very briefly, you might have seen this before in your Bio 21 or Intro Bio course. We have, let's say, purple or violet flowers. This is part of the parental generation. And then we have these white flowers, these pea plants with white flowers. What he found was when he mated these two together, he did not get a blending effect. You would see an intermediate color or an average color, but that's not what happened. All of the flowers here were purple, were violet. And if you've ever studied genotypes, you might have seen big P, small p. Then when he crossed these, the F1 generation, the first filial generation, with other flowers that were in the same generation, in the same genotype, he saw the white flowers reappear again. So these all supported his idea that these traits were inherited through discrete packets, these genes. By 1902, Theodore Boveri and Walter Sutton connected Mendel's principles to chromosome behavior. They observed chromosome movement was important during development and meiosis, and proposed the chromosomal theory of inheritance. And this says that chromosomes are the carriers of genetic material. In the 1900s, Morgan, or Thomas Hunt Morgan, studied or used fruit flies known as Drosophila melanogaster to correlate visible traits with chromosomes. So he studied fruit flies and simple traits such as eye color or wing shape and many others to prove that genes are located on chromosomes. And his 1915 book, The Mechanism of Mendelian Heredity, really cemented this link between visible traits and their location on chromosomes as distinct genes. Barbara McClintock, working with maize, also known as corn, in the 1940s, discovered jumping genes, also known as transposons. These are mobile elements that can move and regulate gene expression. So this explained the patchwork kernel colors in corn and showed that the genome is dynamic. So these jumping genes, also known as transposons or transposable elements, can move from one chromosome location to another in corn. And the movement of these genes actually can turn pigment genes on or off, creating these different colors or patterns in individual kernels on the same ear of corn. So her work really demonstrated that the genome is dynamic, it's not static, and that gene regulation can be influenced by these mobile DNA segments. Hermelin used the giant unicellular alga Acetobularia to show that hereditary information resides in the nucleus. And what he did with these algae was to either remove the cap or remove the foot. And the nucleus is found in the foot for these organisms. When he removed the cap, it was able to regrow because the genetic material was present. But when he removed the foot could no longer regrow. So he also did grafting experiments to support this. I'll show you that in the next slide. In these grafting experiments by Hermelin, what he did was he started working with two different species of Acetobularia. The original one that he worked with in the previous experiment, Acetobularia mediterranea, as well as Acetobularia crenulata. And what he did was he kept the nucleus or the foot intact. He grafted the other species on top of the foot and then wanted to see what kind of cap would be formed in each. And it turned out that whatever was in the nucleus present in the foot would determine the type of cap that was formed. So this showed experimentally that the nucleus was the location of the genetic material, and that would dictate a cell's properties. And just for fun, Acetobularia is also sometimes called mermaid's wineglass or wineglass algae. In the 1940s, Beadle and Tatum exposed bread mold spores from Neurospora crassa to X-rays and found that mutants that were generated because of those X-rays could no longer grow on minimal medium. But by adding specific supplements to the medium, they could identify which step in a biosynthetic pathway was blocked. This led to their famous conclusion or hypothesis that one gene creates one enzyme. Later, nowadays we know that it's really one gene to one polypeptide because some enzymes require more than one polypeptide to function properly. So here's an example of, or a closer look at Beadle and Tatum's experiment. Here are the bread mold spores. We have normal spores and mutants. Mutants were generated through X-ray exposure. They crossed them and then grew them either in complete media, which contains all of those amino acids, or minimal media, which had no amino acids. And they looked for a mutant that grew on complete medium, but not on minimal media. So they looked at this set. Then what they did was they took the mold and they grew it in tubes, basically minimal media, where they only added a single amino acid into the tube, each of these. And they noticed that only when arginine was added into the tube did the mold grow again. So this means that these did not grow because they lacked the production of arginine. And when they examined their mutants more closely, they found 3 different types of mutants. So it turns out that in order to produce the amino acid arginine, which is used in many different types of proteins, and the reason the cells did not grow when they could not make arginine was that because they couldn't make those proteins or enzymes that had arginine as a component of the protein. So to make arginine, there are 3 enzymes involved. It looks like some first enzyme will produce an intermediate ornithine that's passed to a second enzyme, and that will convert it to citrulline, and a next enzyme will convert it to arginine. So that's our final product. So each enzyme, remember, is encoded by a gene. And they hypothesized that there were 3 genes encoding these 3 enzymes. In the table, what they found was that in wild type samples, this is normal mold, it grows on minimal medium because it can make its own arginine. The mutant, however, the first mutant here, cannot grow on minimal media, but does grow anytime any of these 3 are supplied, ornithine, citrulline, or arginine. This shows that enzyme 1 is missing. There's something wrong with this gene. So the X-ray probably messed up this gene, so the enzyme could not be produced. So it can't make ornithine, but it can still use and make downstream intermediates if ornithine is supplied. In the second mutant, so here or here in the table, it cannot grow on minimal media or minimal media with ornithine, but it does grow on citrulline or arginine. This shows that enzyme 2 is missing, so it can't convert ornithine to citrulline. And finally, for enzyme or mutant number 3, mutant number 3 cannot grow on minimal media or minimal media plus ornithine or minimal media plus citrulline, but does grow with arginine. This shows that enzyme number 3 is missing, so it can't convert citrulline to arginine. So what this means is that each gene controls the production of a single enzyme in a biochemical pathway, and a mutation in a gene will block the pathway at that step. But growth can be rescued by providing the product of the missing enzyme's reaction. So this is really the experimental basis of that one gene, one enzyme hypothesis that was later refined to be one gene, one polypeptide. All right, for this checkpoint, they ask, what organism did Morgan and his colleagues use to develop the chromosomal theory of inheritance? So this was Drosophila melanogaster, or fruit flies. And they tracked a bunch of different traits like eye color. Some of the fruit flies had white, others had red eyes. They looked at wing shape and other very easily visible mutations. And this mattered because by following these traits and how they segregated, Morgan was able to link specific traits to specific chromosomes and prove that genes are carried on those chromosomes. For the second one, what did Hammerling prove with his experiments on Acetobularia? He used, remember that giant unicellular algae or alga, Acetobularia. These are huge. These are like anywhere from 2 to 6 centimeter cells, single cells in size. And what he did was, remember, he either removed the cap or the foot of the cell and observed regeneration. He found that only cells with the nucleus in the foot could regenerate a new cap with the original species shape. So the conclusion was that the nucleus really houses the genetic information that controls cell morphology. Moving on to bacteria, in 1928, Frederick Griffith showed that a transforming principle could pass traits between different strains of Streptococcus pneumoniae. So at the time, there were 2 strains, rough and smooth. The R strain, or the rough strain, was non-pathogenic and did not have a capsule on its outer surface, and it appeared rough when grown on petri dishes or plates. In contrast, the S strain is pathogenic and it has a capsule outside of its cell wall. This allows it to escape phagocytosis by the host immune system. So this is often seen in other types of bacteria as well, where having a capsule makes it pathogenic and the non-capsulated version is not pathogenic. So the capsules cause colonies from the S strain or the smooth strain to appear smooth when they're grown on plates. So what he found was that mixing live R strain with heat-killed smooth strain ended up killing the mice and producing live S bacteria. So something from the dead S cells transformed the R cells. And I'll walk you through this experiment in more detail in the next slide. So here's a closer look at Griffith's experiment. He took live R strain, and these were the non-pathogenic version, injected into mice, and nothing happens. The mice survive. If you were to take live S strain, the mice would die because that was pathogenic. But if you take the S strain and you kill the bacterium before you inject that into the mice also survive. What's weird is if you took this one, the live R, and the dead S and mix them together and inject that into mice, the mice die. That's strange, because separately the mouse would survive, so why together would those two cause the mice to die? What's even weirder is when you examine the mouse and examine its blood or tissues, you would find living smooth strain. Like, how did these come back to life? What happened? So they isolated living or live smooth strain from those dead mice, injected that into another healthy host. The same thing would happen. The mouse would die. So what he hypothesized was that something was released from the S cells that were dead and was able to enter the R strain to transform them into the smooth strain. So based on Griffith's experiments, we know that something was leaving the S cells that were dead and entering the living R cells to transform them into living S cells. Today we know it's DNA, but back then they didn't know. So Avery, MacLeod, and McCarty repeated Griffith's experiments with purified extracts, and then they also treated the extracts by one at a time, removing protein, RNA, or DNA. And they found that only when they destroyed DNA, that would block the transformation of live R to live S strain. So they concluded it must be DNA that was the transforming principle. That was converting the live R to live S. And here's a closer look at that Avery, MacLeod, and McCarty experiment. First, they use heat to kill the smooth strain cells, and then they one by one remove different components from these cells. So in the first one, this is the control where everything is intact. Proteins, DNA, and RNA. When they mix the dead S with the living R, they find live S, where the capsule is represented by that outer oval. It's kind of like a grayish cream colored oval. Then what they do is they take the same sample, the heat-killed S, and they get rid of the proteins. So now what's left over is just RNA and DNA from the dead S cells. When they mix it with living R, they again get live S cells. Again, that outer layer is the capsule. Then they do the same thing where they now get rid of RNA instead of protein. So what's present left is proteins and DNA. When proteins and DNA are intact and they mix with living R, they again still get live S cells. In the last experiment where they get rid of DNA, they degrade the DNA and only proteins and RNA are present. When they add R cells, the live R cells, no smooth strains are found. So the transforming principle must be DNA. DNA is turning live R cells to S cells. Okay, so Avery, MacLeod, and McCarty showed that DNA was a transforming principle by taking extracts from heat-killed smooth strain Streptococcus pneumoniae and treated them with enzymes that destroyed either protein, RNA, or DNA. And they found that when DNA was destroyed, transformation of R, or the rough cells, into the virulent S strain or cells did not occur. So when protein or RNA were destroyed, transformation still occurred. So they concluded that DNA and not protein or RNA was the transforming principle that Griffith had observed. In 1952, Martha Chase and Alfred Hershey used bacteriophage T2 virus to infect E. coli bacteria. And what they did to the phage was either label the protein coat with radioactive sulfur or label the viral protein with radioactive phosphorus. And what's really neat is that protein does not contain phosphorus and DNA does not contain sulfur. So by labeling these elements, you could ensure that only one of these was labeled, protein or DNA. What they found was that only DNA entered the bacteria, not the protein. So DNA was what directed new phage production, and this proved that DNA and not protein was the genetic material. So really, it was only after these experiments that the scientific community more broadly accepted DNA as the molecule responsible for heredity. So here's a closer look at the Hershey-Chase experiments. First, they take phosphorus. Remember, phosphorus will label DNA, and there's no phosphorus in the protein shell or capsid for these viruses. And then they allowed the phage to infect the E. coli bacteria. Then they would detach them. So they would kind of blend this and then spin this down, centrifuge this down, and the cells would form a pellet at the bottom. The phage would be in the supernatant. When they examined where the location of the radioactive label was present, they found that the radioactive label was in the cells, but not in the supernatant. So this means that the DNA was injected into the cells. When they labeled this time the sulfur will label the protein coat. They found that when they did the same thing and examined the cells, there was no radioactive labeling. The radioactive label stayed on that original phage capsid. It was not transferred into the cells. So it must be DNA that is the hereditary material. So to summarize, Hershey and Chase used bacteriophage, that was a T2 virus that infects E. coli bacteria, and they either labeled the protein coat with radioactive sulfur or they labeled DNA with radioactive phosphorus. After infection, only the radioactive DNA entered the bacterial cells. The protein coat stayed on the outside. So they concluded that DNA and not protein, DNA is the molecule that carries viral genetic information. And our book has a nice timeline that kind of summarizes all of these experiments, although we haven't talked about some of these on the far right. But overall, by the mid-20th century, a clear picture had emerged, right? We found that DNA is the molecule of heredity, genes reside on chromosomes, and microbes can reveal the biochemical pathways of life. So this really set the stage for understanding DNA's structure and function, and we're going to cover that in the next part of the chapter. All right, let's recap 10.1. For the discovery of nucleic acid and nucleotides, we saw that in 1869, Meijer isolated a substance from white blood cell nuclei. He called it nuclein. And then Altman later found that it was acidic and renamed it nucleic acid. Later, Kossel identified the 5 nitrogenous bases, A, G, C, T, and U, and this will help define nucleotides. And in combination, these steps really established that cells contained distinct acidic substances made of repeating nucleotide units. For the second one, the characterization of DNA, I think this is more relevant to 10.2. So a brief overview for 10.2, we're going to see that Chargaff is going to discover that A's pair with T's and G's pair with C's. And then we're going to also look at Rosalind Franklin and X-ray diffraction for the structure of DNA. They discovered, Franklin and Gosling discovered that DNA has a helix with bases inside and a backbone outside. We're going to see that Watson and Crick built the double helix model. And again, in 10.2, I'll talk more about the DNA's composition, structure, and base pairing. For the third one, how microbiology and microorganisms have been used to discover biochemistry of genes. We talked about Beadle and Tatum, how they used the mold Neurospora crassa to show the one gene, one enzyme hypothesis. Hammerling studied that giant alga, Acetobularia, to localize hereditary info in the nucleus. And then we saw that these experiments led to the discovery of metabolic pathways, gene regulation, and even horizontal gene transfer, basically transferring genes between the same generation. In the last one, how scientists established the link between DNA and heredity, we saw that Frederick Griffith showed a transforming principle, transferring virulence between Streptococcus strains. Avery, MacLeod, and McCarty proved that the transforming principle was DNA and not protein or RNA. And Hershey and Chase used bacteriophage and radioactive labels to show that only DNA enters host cells. So these experiments demonstrated that DNA and not protein carries hereditary information. And that wraps up part 1. In part 2, we'll move into the structure and function of DNA itself.